Hi everybody, so welcome back. This is Sarah Sheffield, your instructor for Geology 3104C at the University of South Florida, Stratigraphy and Paleontology. Tuning in today, we're going to be learning about more clades of common fossil organisms. So this is the second lecture in this series, Meet the Critters 1, introduced us to reef organisms, or common reef organisms like sponges and corals, as well as early life on Earth like stromatolites. So today we're going to be talking about the Lophophorates, which are a large group made up of Brachiopods and Bryozoans, and then we'll be learning about the best group of all, Echinoderms. That's unbiased right there. All right, so we'll learn more about these guys here in just a second. So today we're going to start with the bilaterians, meaning things that are bilaterally symmetrical. So sponges have no symmetry, all right, no organized tissues, and then we move into the cnidarians and ctenophores, which do have symmetry, but it's not bilateral, it's radial symmetry, and that's actually a major change. One of the reasons that we think it is so important for bilaterally symmetrical organisms, because that makes up a huge amount of living animals today, a huge amount. The overwhelming majority is bilaterally symmetrical. The reason that we think that is a reduction in the nervous system, right? Our brains are bilaterally symmetrical. Not many of our internal organs are. If you think your stomach and lungs and things like that, not everything is bilaterally symmetrical on the inside. On the outside, yes, for the most part. Part of the reason we think that is it reduces the amount of essentially brain power that goes into controlling your motion. It reduces it to 2 planes of motion as opposed to, like a jellyfish, many planes of motion, which allows us to move with more efficiency. So, something to think about. Pretty cool. All right, so today, again, Brachiopods, Bryozoans, and Echinoderms. This phylogenetic tree does have some differentiations if you look at it in other sources, but this is the general idea here. So we're going to skip around just a little bit, but we'll focus on this group today and the Echinoderms, and we'll come back to other major groups like the mollusks and arthropods a little bit later. So bilaterians, again, animals that have twofold symmetry. There's 2 groups within it, the protostomes and the deuterostomes. We're not going to worry too terribly much about the proterostomes and deuterostomes. I'm going to flip back really quick to show you where those are. Protostomes are over here with our friends, the arthropods, bryozoans, brachiopods, mollusks, etc. And echinoderms, excuse me, and chordates, that includes the vertebrates, we're over here with the deuterostomes. Really, this has to deal with how our embryos develop and where our mouth and our anus is forming from. So in protostomes, the mouth forms from the blastophore, and in deuterostomes, the mouth is forming in a different area up here, and the anus is actually forming from the blastophore. We're not going to worry too much about the difference between these two for now, but we're going to go through the major clades here. So the first are the Lophophorates, things that use lophophore feeding structures. The first of which are the Brachiopods. These are an incredibly important clade of organisms. These are almost exclusively Paleozoic, although they are alive today. Just the 99.9% of them were around in the Paleozoic. They have 2 valves, shells, a lophophore for feeding. This is filter feeding. How this works is it creates a teeny tiny little current that kind of draws little particles to it. And their bilateral symmetry is through the hinge. What this means is, unlike clamshells, if you take a look in your fossil kit, clamshells, each shell as a separate piece is identical to the other one for the most part. And brachiopods, you'd have to cut the organism in half. Instead of each shell being the same, it's literally straight down the middle. So brachiopod literally means arm feet. They lived from the Cambrian and they are still alive today. However, they took an extremely heavy hit in the Permo-Triassic mass extinction, and they never really recovered. If you and I were to take a stroll down the Paleozoic beach, these are the types of organisms we would see as opposed to the snails and clams we see today. They're made of either calcite or calcium phosphate. They're exclusively marine. And as I said earlier, they filter feed through a specialized organ called the lophophore. We'll talk more about that in just a little bit. There's 2 major groups, the articulates and the inarticulates. The articulates are going to be the much more common fossils. Inarticulates, as you can see from this bottom picture, are still alive today. This is a modern organism. The articulate ones are much more diverse than the inarticulate ones. So the differences between them are that the articulates have hinges. That's what they're articulated. That's where the name comes from. They have hinges to open and close. They have teeth, little ones in the back, right where my cursor is pointing here. We'll see those in a little bit. And that help keep the valves closed tightly. These are made of calcite. Inarticulates, on the other hand, don't really have hinges. They actually have muscles to open and close, much stronger muscles because they don't have those hinges to rely on. And these are made of calcium phosphate. So this means that even though there's not a whole lot to these critters, we actually do have a lot of these in the fossil record because if you'll remember with me, calcium phosphate is an incredibly powerful fossil-forming mineral, the same fossil-forming mineral that makes up bones and teeth. So here we have our brachiopods through time. As you can see, they are extremely diverse. Here are the different orders: Orthida, Strophomenida, Pentamerida, Rhynchonellida, Spirifida, and Terebrachiopoda. Try saying that 5 times fast. All right. They're facies dependent. However, they're still really important stratigraphic fossils in the Paleozoic record. And the reason for that is that they are globally found. They are extremely common. They are easily identifiable. So there's a whole lot of really great, and they evolved quickly. So really great fossil record here for biostratigraphy. So here's a really good example of when they're extremely important. However, we can see right there at the Permo-Triassic boundary, holy bananas, do they really take a hard hit, and they never really recover. Terebrachiopods are really the only ones that exist in any kind of excessive realm today in the articulate world. Brachiopods do appear to be zoned according to sediment grain size. At first, we thought it was depth and potentially related to oxygen levels. However, it does appear to be more related to sediment grain size. So here's how this works. All right, each of these colors here aligns with a certain depth. Remember, grain size and depth are related via water energy. The larger grains will likely be settling out closer to the coast, whereas the deeper in the sea we go, we typically have finer grain material like silt and clay-sized grains. There are some small differences between the two, but we're not going to worry about that right now. Brachiopods also show incredible ecophenotypic variation. That is a $5 word. Eco meaning ecology, phenotype meaning your outward appearance, and then variation meaning differences, right? So differences based on, differences in how you look based on the ecology around you. So how this works is here we have 3 different types of brachiopods: spirifers, atrepas, and terebrachiopods. Each of these 3 types can actually look extremely different depending on the increase in hydroenergy. What this means is if we don't know better in the fossil record, we might actually categorize these 2 as different species, or even these 2 as different species, even though they're the exact same. We learned this on our Critters 1 assignment, where corals are really great at ecophenotypic variation. Many organisms are. What this means is that Brachiopod taxonomy, identifying the species, will have to look at the internal morphology and the shell microstructure. For our purposes, though, we are looking at the majority of what we're looking at are pretty easy to define species. So here's what our internal anatomy looks like. We have the 2 valves on our brachiopod. The pedicle valve, which is always the larger valve. So if you're not sure which is which, whichever one is the larger one is the pedicle valve. At this point, please pick up your fossil kit. All right, open it up. Pull out your brachiopods. They are going to be located on the left-hand side, closer to the back, if I remember correctly. So pick up those, look at the pedicle valve. It is always the larger one. If you flip it over, you should see a little mark where the pedicle, which is a soft tissue piece that kind of works like a foot, it would stick out so it could stick to the hard substrate. This is a drawing of what it would look like in life position. The brachial valve is always a smaller one. Inside, there would be the lophophore. This is the lophophore right here. All right, now this says C demonstration. That's a holdover from when, people did in-class exercises. So what I want you to do is take your arm, curl it up close to your chest, and then splay it out kind of like, let me think here. It's kind of like you're throwing a bowling ball, but flip it down instead of up. All right, so it's kind of going to look like that. The lophophore kind of unfurls from the organism and it creates a little microcurrent by using these little hairs to draw in little bits of food. These organisms are classically low in metabolism, meaning they don't really need a whole lot of energy. Here is more of the internal anatomy. I don't need you to memorize any of this, but this is what it looks like on the inside if it were still alive. It has a shell opening and a shell closing muscle. One of the things that is interesting about this is that unless the muscles, when they're not activated, they actually just remain closed. This is opposed to bivalves, on the other hand, or many bivalves. I shouldn't say all bivalves. So that's one of the reasons that we typically find brachiopods found together in the fossil record, is that when they died, those muscles are actually closed. So here's the outside part of that shell. Here we have our pedicle valve. It is the larger of the valve. This is the brachial valve, the smaller valve. Here's the pedicle opening, which will always, of course, be found on the pedicle valve. There are different types of features that we can see. For example, the deltherium, which is a triangular, that's where it gets a delta from, triangular. A little bit right there underneath the pedicle opening. All right. We also have the hinge line, which is right here. We have the beak, which is the sharp point on it. We have the sulcus. That's the fold that we see on a lot of them. A lot of our brachiopods will have little folds on them. Please pick up one of your brachiopods. See what you can find. Look at the hinge line. One of your brachiopods, the larger ones, you have 2 or 3 containers. One has 10 brachiopods in it. The others have 2 or 1 of each. There's one with an extremely large hinge line. You are either looking at Raphinaskina or Leptana, genus names. It has a very large hinge line. You have strong sulcus on many of those in your little box containing 10. You have folds. Some of yours have very strong plications. These are lines across your organism. All of these vary greatly from species to species. As I said earlier, unlike bivalves like clams, the shells themselves, the 2 shells, are not symmetrical. The valves are asymmetrical. Again, the pedicle valve is larger, and that fold and sulcus gives each valve very different looks. So the symmetry actually goes straight through the organism instead of between the valves. Brachiopods can give us a lot of clues about how they used to live based on the different types of morphologies that we see. I'm not going to worry about this too much yet. We'll learn about this a little bit more in our in-class assignment and during our lab. But the different types of features show us a very different lifestyle. For example, the larger the pedicle opening, the stronger pedicle attachment to the surface needed, probably meaning that they lived in higher energy environment. The same with strong ribs or strong plications, those lines running across the sediment or across the organism. That would indicate that they're living in high energy water currents, so those lines actually break up the water current coming across the valves, which is something we actually see happening in multiple different groups of organisms. So it's a convergent type of feature there. So there's lots of different ones. We'll talk more about these in the lecture themselves, but this is where you get that information from. So let's now talk about the moss animals, the bryozoans. Now, bryozoans get a bad rap for being boring in the fossil record, but they're super cool, especially if you own a scanning electron microscope. However, very few of us do, of course, own a hundreds and hundreds of thousands of dollars scanning electron microscope, so we're just going to have to rely on photos here. Here is the bryozoan extending its lophophore, just like the brachiopod. Now, you might be thinking that brachiopods and bryozoans look nothing alike. You'd be correct. But they have this incredibly important lophophore in common, the synapomorphy linking them together. Really cool, right? You just want to keep watching it. I know. So here are what bryozoans look like under SEM, which is so cool. All these little holes are actually corresponding to different zooids. Bryozoans are colonial, meaning there are dependent animals living inside the system, kind of like a community. They're called zooids because they are not independent from one another. Each one has slightly different functions. So you have some that are in the mode of feeding, some that are in the mode of respiration or reproduction or defense, things like that. Some actually have incredibly sharp spikes on their bodies so that they can actually defend themselves against organisms trying to snack on them, which is super cool. They're from the Ordovician to recent. They are the only known phylum that did not show up in the Cambrian explosion that we know of. They're made out of calcium carbonate, largely calcite. Pretty much all marine. Everything except for one genus is colonial, and they have very itty-bitty little zooids, no septa. That's how you tell them apart from corals. They don't have those little lines in them. Lots of different ecophenotypic variation. Lots of different types of bryozoan colonies. So some can be branching or sheet-like or encrusting. Most of our modern stuff is encrusting, like this little critter here. It is encrusting on what looks like just a boring pebble. All right, here are some branching ones. We'll look at some other forms here in just a second. So as I said earlier, different zooids, the little itty-bitty individual organisms, take on different roles to support the colony itself. These appear to be the greeters of the colony. I am kidding, but they do make some excellent faces, right? Here are some that actually will act as barriers for protection. Some look like these are fenestrate bryozoans. They're not necessarily encrusting. These are like fan-shaped. Fenestrate means window in Latin, which is where it gets its name from. This is the typical fenestrate shape, right? These little windows here. You have an Archimedes in your fossil box. Comes from the name for Archimedes screw, the invention that Archimedes built to get water from an area upstream. Looks just like it. If you zoomed in with a microscope and looked at the microstructure, you would see this exact fenestrate pattern on it. We have our branching bryozoans. Here they are. You'll notice that there's different types of bumps all over it. The bumps are called monticules, M-O-N-T-I-C-U-L-E-S. The zooids live on top of little holes in the monticules. Here's an encrusting one. What is it growing on? You have an exact fossil of this in your kit. I hear you shouting Brachiopod. Yes, you are absolutely correct. This is a Brachiopod it is growing on. Very nice. Look at that beautiful hinge line. Giant hinge line. Now, one of the problems with bryozoans is that we can't identify them well in hand sample. There's a couple really common ones. For example, Archimedes, the screw shape, is extremely identifiable. It is in fact a famous index fossil living in the Mississippian. So if you ever find one, you know exactly where you are in the fossil record. But most of the time, to tell down to the genus and the species, you have to take thin sections of them and study them for micro differences. They are absolutely beautiful in thin section. They look more like stained glass, don't they? All right, picking up with the next video, we will be talking about echinoderms. See you in a bit.